Fridays at 8:30, so we're celebrating vision research. Thank you again for all you joining by web and here in the room. Today we have E.J. Chichilnisky, the guy who really practiced real science, which is math. And E.J. made a lot of wonderful contribution and fantastic papers. And hopefully, again, we will learn something. That's the idea today from our speaker. So without any delay, Emily.
Good morning, everybody. Thank you for joining us for our seminar. My name is Emily Tom, and I'm a fourth-year MD student in the Retina Club. And it is my great pleasure to introduce today's distinguished speaker, Dr. E.J. Chichilnisky, John R. Adler Professor of Neurosurgery and Professor of Ophthalmology at Stanford University. Dr. Chichilnisky earned his BA in Mathematics from Princeton University, his MS in Mathematics, and his Ph.D. in Neuroscience from Stanford University. Prior to joining the faculty at Stanford, he worked at the Selectivity Section of Biological Studies for 15 years. Dr. Chichilnisky's research has focused on understanding the spatiotemporal patterns of electrical activity in the retina that convey visual information to the brain and their origins in retinal circuitry using large-scale multi-electrode recordings. Currently, his lab is integrating basic science knowledge along with electrical information to develop a novel high-fidelity artificial retina for treating incurable blindness. To achieve this goal, his lab works closely with collaborators in fields spanning neurophysiology, electrical engineering, material science, retinal surgery, visual behavior, and computational neuroscience. And together, they make up the Stanford Artificial Retina Project. Dr. Chichilnisky has made tremendous contributions to our understanding of the retinal circuitry and how it encodes visual information. He is the recipient of an Alfred P. Sloan Research Fellowship, a McKnight Scholar Award, a McKnight Technological Innovation in Neuroscience Award, and an Einstein Innovation. In addition, he holds several patents for his innovative technologies. And it is our pleasure to welcome you here today. Please give a warm welcome to Dr. E.J. Chichilnisky, who will be presenting his talk entitled Toward a High-Fidelity Artificial Retina.
So Tony, you can hear us very well. Anand, can you hear us well?
Can you hear me well?
Not really very well. I have to, yeah, sound is really not very good.
Okay, so let's take a microphone or podium microphone. 1 second, guys, sorry for that.
No, I even put on my headset and the sound was breaking up and her voice did not carry well. Yours carries pretty well.
Can you hear me well now?
Oh, yes, beautifully.
Yeah, now much better.
I would turn it all the way down in this room so I can speak. There we go. Does anybody in the room have trouble hearing me?
No.
Okay, and the remote audience, good now?
Yes, pretty good.
Yes.
Yeah, you sound good, EJ.
You can hear that's the mic.
Perfect.
Oh.
You sound great. Perfect.
Yes, you do.
Somebody give a thumbs up.
Anand, are we good?
Yeah, you're good remotely.
Good. Okay, you got a stamp of approval.
I got the stamp of approval. Thank you all for coming today, and thanks to the Zoom participants. I didn't realize you had such a broad audience on Zoom, which is fantastic. I really appreciate the invitation to come here today and the kind hosts who have made it very fun for me already, actually meeting everybody. So thank you for having me. So it's my pleasure to talk to you today about our research on the development of a high-fidelity artificial retina, which I will define as an implanted device that will faithfully reproduce the neural code of the retina. So let me give a little brief outline of what my talk will be today. First, why would we want to develop an artificial retina? Second, what are the most important aspects of the neural code of the retina that we would need to emulate to do this? Third, I will tell you how we can test our ability to reproduce the neural code of the retina using an experimental lab prototype. Then I'll tell you a little bit at the end about our project developing an implantable device that will do the same thing and the possible future implications. Okay, so first, why would we want to develop a device like this that would reproduce the neural code of the retina? And the obvious answer is shown here, namely that visual disorders that render people incapable of natural visual function are obviously very life-changing. And although we have some excellent technologies that are old and some newer technologies as well, ultimately we might hope to be able to actually restore vision rather than to replace it with different kinds of modalities. But there's a second reason too, beyond this is enough reason for me to focus on this project, but there's another reason as well, which is sort of a scientific and philosophical reason, which is the retina really is arguably the best understood nervous system structure, with decades of research trying to understand everything about it, from how it captures light and processes it to the neural code that sends information to the brain. If there's any part of the nervous system that we might be able to effectively replace with implanted devices to instantiate our scientific knowledge in actual development of devices to help humanity, the retina is the first place to do that. I believe in that passionately, and I think the time has come to develop such a device. So it's really an ideal test bed for understanding and our neural signaling and figuring out whether our understanding really translates into something that can help people in a day-to-day way. So the concept of an artificial retina or something like that is obviously familiar to many of you, and I'll just discuss it briefly. As you know, in some blinding diseases, notably age-related macular degeneration and retinitis pigmentosa, the photoreceptor cells of the retina are lost for various reasons, rendering the retina unable to transduce light into electrical signals and thereby producing blindness. So, however, significant parts of the neural circuitry remain present and even to some degree intact, notably the retinal ganglion cells that transmit visual information to the brain. So this opens up the possibility of a technological intervention illustrated schematically here, namely a camera that captures the visual image, processes the signals from the camera appropriately, and then electrically stimulates the remaining retinal ganglion cells, causing them to send artificial visual signals to the brain. The easy part is the camera. The hard part is the processing and stimulating of ganglion cells, which is what my talk will be about today. Now, obviously, we're far from the first people to think of this. In fact, the best known work in developing a device of this general character is the Argus 2 implant developed by Second Sight Incorporated in the Southern California area. A schematic of this device is shown on the left with the camera that captures image, the processing unit, and the electronics for electrically stimulating the retina. And this is the array of about 60 implanted electrodes on the surface of the retina, a couple hundred microns in diameter each. So as many of you know, this device on the one hand was successful in the sense of eliciting visual perception in patients who've been profoundly blind for decades. And that's a very exciting development that we can do this. On the other hand, the perception that was evoked was crude and ill-controlled and not very useful for visual function, unfortunately. And I think it's well summarized by this quote in the New York Times on the day that this device became commercialized for use in the United States. With it, people with certain types of blindness can detect crosswalks in the street, burners on the stove, the presence of people of cars, and even sometimes oversized numbers or letters. Detect, I want to emphasize detect. So with this device, you can know that something is there. You might see some crude outlines of it, but you're not really having the kind of visual experience that we hope for, which is recognizing complex objects, navigating novel environments, appreciating beauty, and appreciating emotions on faces, and all that. None of that is available with these kinds of devices. So why is that? And why is this device no longer actually sold in the United States? And what would it take to move forward? So obviously 60 coarse electrodes on the surface of the retina is unlikely to deliver a visual image that would be of high interest to us. And so the idea of just more electrodes in order to deliver a richer visual image is obvious, and people have worked on that and continue to work on that, including us. But there's a deeper reason, I think, for why this device and similar devices that are similar to it have not been delivered really outstanding visual perception to restore natural vision. And I think that is summarized by looking at the retina itself. The retina is a complex neural circuit, as you know, with photoreceptors that encode the visual signal, many different types of interneurons that process the visual signal, and then the retinal ganglion cells that send visual information to the brain. There are dozens of distinct circuits in the retina and at least a couple of dozen distinct types of retinal ganglion cells. And these different types of retinal ganglion cells differ in fundamental ways. They differ in their morphology, they differ in their connectivity to other retinal interneurons, they differ in their light response properties from one another, and they differ in terms of where they project visual information to the brain. Furthermore, each of the different ganglion cell types transmits a complete representation of the visual scene to its particular targets in the brain. So we really want to think of the retina certainly not as a simple camera or something like that, not even as a neural circuit that processes the information, but maybe as 20 distinct neural circuits that process the information in many different ways and transmit the results to a variety of targets in the brain that ultimately give rise to our visual experience. So this is a complex device, and replacing it with a grid of 60 electrodes, or attempting to replace its function with a grid of 60 electrodes that has no relationship to all of this circuitry and processing, seems potentially problematic. And that's the issue I want to address today. So the guiding hypothesis is that the neural code of the retina actually matters. These distinct signals transmitted by the distinct ganglion cell types to their variety of targets in the brain are important for vision. And in order to really restore high quality natural vision, we're going to need to be able to emulate that neural code. That is the guiding hypothesis of my talk today. So what I'm going to do is go into just covering a little bit of the basic research that gives us some sort of signposts for what aspects of the neural code we think we need to replicate in order to create high quality vision. And then a collection of experiments to show that at least to a pretty good degree, we can replicate many of those signals. So let's do the basic science first and cover what are the key aspects of the neural code. Well, to do that, of course, we need to think about animal models for visual function, and there are multiple animals that are used for vision research. This one for high acuity vision, this one because of its ease of use. I don't have to explain why people use mice, but it's certainly not for their acute human type visual systems. Then there's the macaque monkey, and we focus on the macaque monkey retina because it's the most similar to the human retina. That's the reason. It's a difficult species to work with in a variety of ways. We obtain macaque monkey eyes from other labs that are using the animals in the course of their research. So when the animal is euthanized, we collect the eyes, bring them back to our lab, and perform experiments on them for 48 hours or so nonstop in order to collect the kind of data I'll be telling you about in the next couple of minutes. I want to emphasize that the data I'll be telling you about are all from my lab. That's not because we're the only people who have done that kind of research, but it kind of synthesizes a collection of work in the field, in the macaque monkey, that in a simple way that I think makes the points I want to make. I will also point out a couple new instances where we're performing experiments on the human retina from donor humans as well. Okay, so to understand these basic aspects of the neural code, we perform large-scale multi-electrode recordings from these excised pieces of macaque retina using a preparation shown here, where a small piece of retina is placed flat on a grid of microelectrodes that record activity extracellularly from retinal ganglion cells. The retina is perfused with an oxygenated saline solution and stimulated from above with the optically reduced image of a computer display. So we're providing a naturalistic input to the retina while recording the output of many different types of ganglion cells. We use custom large-scale multi-electrode recording arrays for doing this with 512 electrodes in various different geometries and a pitch of 30, 60, or 120 microns. This allows us to record from lots of cells, and you'll see in a minute why that's important, all in a single retinal preparation while stimulating it with light. When we do this, we characterize the cells typically using so-called white noise visual stimulation. For those of you who haven't seen it yet, I'll summarize it briefly. I skipped over talking about the voltage recordings that we obtain and the identification of spikes, which is necessary in these circumstances, but I won't go into that in detail. So we have to separate the spikes out from the many cells we record. So white noise visual stimulation is shown here, where an image that consists of a random checkerboard pattern updating randomly over space and time is displayed to the retina for tens of minutes. We then collect up the spikes from the hundreds of cells that we record from simultaneously and analyze them. And for each cell, we identify the spikes that were evoked by the white noise visual stimulation and perform reverse correlation with the stimulus to figure out what aspects of the stimulus were important for driving the spikes. So conceptually, we go to each spike, we figure out what was the stimulus sequence in the, call it 250 milliseconds before that spike, and and we take note of that, and we go to the next spike, we figure out what happened before that spike and take note of that, and so on through all the thousands of spikes recorded from a given cell. Take the average stimulus that preceded a spike, and that's the so-called spike-triggered average stimulus. That stimulus is a brief little movie clip that I'll play at the right now. What you can see is that about 50 milliseconds before firing in this particular cell, there tended to be a brightening in a particular region of the display. We refer to that as the receptive field of the cell using conventional nomenclature, and we can summarize it with, let's say, an ellipse like this. And we can also understand the temporal properties and chromatic properties of the cell by looking at the average spike-triggered average stimulus in that location of the visual field. So this is the characterization, the simple characterization of the properties of a single ganglion cell, but we're recording simultaneously from hundreds of ganglion cells. And the advantage of that, I think, will become clear in a second, which is that we can use the spike-triggered average stimulus to figure out what all the different cell types are and what they're all doing simultaneously in one little region of retina. An example of that kind of analysis is shown in this slide, where we've clustered the hundreds of different cells from a single retinal recording shown here according to properties of the light response characterized in the previous slide. And when we do that clustering, we can separate out the different cell types because they form these distinct groups, and see that their receptive fields form a very beautiful uniform tiling of the visual space over the multi-electrode array. So these are 7 different cell types, all in the same retina, all on top of one another, but they've been segregated out according to their light response properties. The fact that each of these different groups of cells forms a tidy uniform mosaic is very important, because we know that each of the different ganglion cell types forms a mosaic over the surface of the retina. So these cell types must correspond to the known morphological cell types in the macaque and the human retina, which are very similar to one another. We can also see that there's a distinct type of temporal properties and chromatic properties of light response, as well as autocorrelation indicative of the firing properties of the cell over time. So this characterization then in a single retina gives us access to, in this case, 7 major ganglion cell types, which constitute about 75–80% of the entire visual representation transmitted to the brain. Notably, the on and off parasol cells and the on and off midget cells that are some of the best known for cell types, and their properties such as their spatiotemporal and chromatic response properties. As a brief aside, I just want to mention that exactly this kind of representation in terms of all the different cell types and their properties and all that shows up beautifully in the human retina when you perform human retina recordings as well. So this is highly applicable to human vision. Okay.
Can I ask a short?
Yes.
It's a peripheral region, right?
This is peripheral retina. I will address that later in the talk.
Thank you.
Yep, that's a super important question. Most of my talk will be peripheral retinal recordings at 6 millimeters and beyond, 6 millimeters and up. But toward the end of the talk, I'll address moving toward the fovea. Okay, so these are our recordings, and with these recordings, we can see all the properties of all different cell types, all in one retinal preparation, all simultaneously recorded, how they cover the visual scene and all of that, okay? Now, what are the main points of the neural code of the retina that we really need to think about in terms of reproducing the capability of the retina to send visual information to the brain? I will focus on the following few properties. First is the exquisite temporal precision of the signals delivered by retinal ganglion cells to the brain. If you take a single retinal ganglion cell, such as the one that's shown recorded here from the macaque retina, and you provide a visual stimulus, in this case, a pseudo-random flickering checkerboard stimulus, doesn't matter what it is, what we see is that the patterns of activity are highly reproducible from trial to trial. So each tick is a spike, each row is a repeat of this visual stimulus, and this cell is doing basically the exact same thing over and over and over again. In fact, the temporal precision of the spiking can sometimes be as high as about a millisecond. So the variation in the timing here is about a millisecond. So the neural code of an individual retinal ganglion cells is exquisite and precise. More importantly, not only is just one cell exquisite and precise, but each cell type has its own very predictable, precise neural code. And we can see that by looking at multiple cells of each type in the same recording. So here's a so-called on-parasol cell, one of the major 4 ganglion cell types in the retina, provided with a visual stimulus, gray, white, gray, dark gray, gray. And the spikes are shown overlaid here for simplicity. You can see that this particular cell fires at these particular times in the stimulus cycle. Okay, great. That's one cell in one recording. Now, if we go to another on-parasol cell in that same recording and provide it with the same visual stimulus, we see it's basically a little carbon copy of that first cell, for those of you who know what a carbon copy is, okay? So it's nearly identical in all of its properties to that first on-parasol cell. Now we go to a different cell type, let's say the off-parasol cells, and no surprise, it's a radically different visual signal transmitted by that cell to the brain. If we go to a second off-parasol cell, again, a little copy of the first one, and so on. As we march through the different cell types, not only is every cell incredibly precise, but each cell type has a very particular type of dynamic visual signal that it transmits to the brain. We now have a pretty good understanding of these multiple cell types through the experiments of many labs, including my own, to understand the major cell types in the retina. And these properties that are exquisite and precise in each ganglion cell type can be understood pretty well using models of this form. I'm not going to go into the detail of the model in the interest of time, but many models have been developed of retinal signaling. And in many cases, we can predict the responses of retinal ganglion cells quite accurately using models of a relatively simple form to a variety of visual stimuli. So we're in a pretty good place in terms of understanding what's the neural code that we need to reproduce in order to deliver the correct visual signals to the brain. This is the image that I hope we carry around, therefore, about the retinal signal. Complex patterns of activity in each of the different ganglion cell types, totally different patterns of activity in the different ganglion cell types, transmitting the visual representation to the brain. The challenge, of course, is that these different ganglion cell types are not neatly separated out from one another for us to access separately. In fact, they're all mixed up on the retinal surface. So if you come in with an electrode array, such as the second sight device or any other, and you simply stimulate according to the intensities of the pixels in the image, you cannot create these distinct patterns of activity in the different ganglion cell types that the brain is expecting to receive. Instead, you're going to basically make them all do the same thing as one another, which never happens in natural vision. Okay, so that, in my view, is the central challenge of reproducing the neural code in the retina, which is that all the different cell types are doing different things, and we somehow have to address them distinctly. What's that going to take to address them? Well, let's imagine what a device would be that would reproduce the neural code of the retina. And I'll give you what we think of as such a device. We think of an artificial retina. What are the three things that a device like this needs to be able to do in order to properly reproduce the neural code once you implant it in a retina? The artificial retina that we conceive of will do three principal things. First, it will record spontaneous activity that's present in the retina without light stimulation and use those recordings to identify the distinct cells and to identify the distinct cell types. You'll see that information in a minute. Second, this device would stimulate and record in order to figure out which electrodes activate which cells of which types by how much. These we can think of as two calibration steps. Figure out what the circuitry is, and then figure out how we can control it with the device that we've implanted. Once we have all that information, and we've calibrated this thing to within an inch of its life, we envision saying, okay, great. Now we have an incoming image. We know how our device activates the different cells and different cell types on the surface of the retina. Let's go and reproduce that visual image as accurately as possible. So I'll be now telling you about a sequence of studies designed at doing these three things together. And then finally, I'll mention the development of the implanted device itself. So what do we need in order to be able to build a device that does these kinds of things? And I'm going to talk today about some of these necessities. So first of all, we have to be able to electrically stimulate at something like single cell, single spike resolution. Single cells, because if you stimulate two cells, they're two different types, and you're going to make a mess of the neural code. Single spikes, because it seems like every spike matters more or less, given the incredible precision of the neural code. Second, we of course need to be able to evoke spatiotemporal patterns of activity in many cell types. We can't just activate one cell, we have to activate patterns of activity. Turns out we need to detect and avoid unwanted axon activation, which can make a real mess of the neural code. We'll talk about that. And of course, we need to target areas in the central retina for doing this. I won't mention the last two types in my talk today in the interest of time, but I will tackle the first four, and I'll tackle them in the context of this exact same laboratory prototype that I've been telling you about. And then at the end, I'll mention the idea of building an implantable device. Okay, so let's go to these four topics. First, electrically stimulating at single cell, single spike resolution. We use the same experimental preparation for doing this with different electronics that allows us to pass current through an electrode while at the same time recording the patterns of activity on many electrodes. And the key here is that because we've recorded these complete populations of all these different cell types, we really know what's going on in the retina when we pass current through a given electrode. We're not just recording from one cell with our electrode and not knowing what the rest are doing. No, we're recording from basically all of the cells of all the major cell types in that region, and we know exactly what that electrode is doing to one cell or multiple cells. We typically pass triphasic charge balance current waveforms, such as the one shown here, that are on the order of a microamp of current delivered for a tenth of a millisecond, 100 picocoulombs of charge. This is a tiny amount of charge delivered to the retina, tiny compared to previous devices. That's actually fairly important. I won't go into it in detail. Let's see what that looks like on an experiment. So here's an experiment where we're showing voltage recording from a particular electrode while stimulating with a neighboring electrode. In this case, we pass an electrical stimulus at this moment in time. What happens is after we pass the stimulus, there's a voltage deflection shown here. That's the same on every single trial that we do this. However, shortly after this primary voltage deflection, which turns out to be a simple electrical artifact, because you pass current in one electrode, record from another, you're going to get some recording of that electrical stimulus. But after that, we see the curves diverge in different trials into two different sets of curves. And we call these successes and we call these failures, because if you take the average of these and you subtract the average of these, what you get is a beautiful action potential waveform. Not only does it look like a perfect action potential, but it has the exact same shape as action potentials that are recorded in spontaneous activity on that same electrode. So what's happening is that in roughly half the trials, we're eliciting a spike in this cell at this time, and in the other half of the trials at this current level in this electrode, we're getting no spike from this particular cell. As I mentioned, the charge densities are tiny and well within established safety limits. Also note the temporal variation here. This spike is being evoked at a very precise time relative to the electrical stimulus. That's because it's direct. We're directly evoking spikes in the cell. There's no intermediate circuitry involved here. With a tiny amount of current, we're depolarizing the cell directly and causing a spike in the cell. And we've confirmed that with experiments in synaptic blockers. The electrical stimulation thresholds are again very similar in macaque and human. So these approaches that I'm telling you about for reproducing activity in the retina apply very well to the major cell types in the two species. Importantly, we can address all the different cell types that we see with this electrical stimulation. So this is an example of showing the separation of spikes from no spike in the 5 major ganglion cell types of the macaque retina. We see that as we increase the pulse amplitude, it's systematic. The probability of a spike in the cell systematically increases from 0 to 1, as we would expect. The temporal variation of the spike that we evoke is tiny. It's on the order of a tenth of a millisecond, much more precise, in fact, than the natural neural code. And perhaps most importantly, we can frequently stimulate a single cell of a particular type to fire a spike with sub-millisecond time resolution while not activating any of its neighbors of the same type. And that's shown here for each of the 5 major ganglion cell types in the macaque. Okay, so the spatial precision can be exquisite. Now you may be wondering, wait a second, if I activated that cell there and I didn't get any of the neighboring cells of the same type, did I activate one of the other cell types at that location? That experiment is shown here with 4 major cell types, all on top of one another, in fact, but separated out for your visualizing convenience. What we've done is target a particular cell for electrical stimulation, use a particular electrode, passed a particular amount of current through that electrode, and caused a spike in this cell with probability close to 1 without evoking spikes in any of the neighboring cells of the off-midget type, the most dense in the retina, or the other 3 major ganglion cell types. This, I want to point out, is the ideal in terms of reproducing the neural code. We're able to evoke a single spike in a single cell with sub-millisecond time resolution. If we could do that all the time, we're done with that aspect of controlling the retinal signal. We can't do it all the time, and I'll spend some time on that in the next few minutes, but sometimes we can. Other times, we might target a particular cell for stimulation. We're able to evoke activity in that cell, but we might get a little bit of splatter onto a nearby cell. Other times, we have an even harder time where we try to evoke a spike in a particular cell. We're able to do that, but unfortunately, at lower current levels, we activate a spike in another cell. So this is the kind of range of stuff that we're seeing. Importantly, I want to distinguish this. It's not perfect. We can't get every cell to fire a spike exactly when we want it to, but it's a far cry from typical electrical stimulation where you activate dozens or hundreds of cells with unknown timing and reverberant neural activity. This is very precise dialing up of the neural code. And it's close to what we want to be able to achieve. As a summary of the selectivity, I'm showing you data here from 8 different retinal preparations, looking at the on and off parasol cells, 2 of the major ganglion cell types, the selectivity metric, the ability to stimulate a single cell without stimulating any of its other types of on or off parasol cells in that particular retina is shown here. There's a lot of red in this, which indicates we frequently can activate these cells, the on and off parasol cells, very precisely, while not activating any other cells. So that's a pretty good setup in terms of reproducing the neural code.
EJ, if I may just,
Yeah.
since we're moving to a little bit different topic.
Yep.
Would it be possible, a little bit more difficult experiment, this one is sufficiently difficult, but to stimulate from like bipolar cells or photoreceptors, you still have photoreceptors in this case, but if you wouldn't, then go to bipolar cells and to see whether your down in ganglion cells input is the same.
Yeah, the short answer is that's much harder, as you said. I would rather answer that at the end because it will take us pretty far afield, but please bring that question back at the end. Because that's,
If I will remember, I will.
if you, okay, we're going to try to remember together. Okay. Okay, so this shows that we can frequently electrically stimulate at single cell, single spike resolution. Now, that doesn't help us very much if all we can do is get one cell at a time. We need to create patterns of retinal activity in order to reproduce the neural code. Can we do that? So a nice neural code example that we try to emulate is this one here, which is the light evoked activity of a collection of, in this case, on paracel cells that's produced by the movement of a bar across the retina. What you can see in this movie, I hope the Zoom viewers can see it as well, is a pitter-patter of background spontaneous activity followed by a wave of activity that's produced as the bar moves across the retina. This is an example of some kind of pattern of activity we would want to create in the retina. Can we do this? Yes, we can. Here's an example of that in a particular experiment. This is a complex experiment because it involves the following steps. First, we take this bar of light, we sweep it across the retina, and we record the patterns of activity, in this case, in these 6 cells. Then we go and calibrate the system, as I told you, stimulating through every single electrode, analyzing the responses of all the cells to each of the different electrodes at each of the different current levels. And then we go back and, having chosen the correct electrodes and correct current levels, attempt to reproduce this pattern of activity in this small collection of cells. So we pass current at these particular times recorded here, and here is the evoked electrical activity that we obtain. What you can see is that the black dots and the gray dots are very well aligned. In other words, we're able to reproduce those patterns of activity in this collection of cells with very high fidelity. So again, this is in the direction of being able to really reproduce the neural code at high fidelity. Okay, so we're able to evoke spatiotemporal patterns of activity. As many of you know, however, there's an additional challenge, which is evoking patterns of activity through the axons, which is something we don't want to do. This is an image we took in our lab of the axon and ganglion cells on the surface of the peripheral retina. The cell bodies are shown here. These bundles are bundles of axons coming from further parts of the retina crossing the region where we are. Now, unfortunately, if we put an electrode array up against the retina, our electrode array is basically closer to the axons than it is to the cell bodies. The cell bodies are actually further in from the retina. That's a bit of a nightmare, as you can imagine. And if we get lucky, we might be able to have an electrode that lands right smack on a cell and is not on an axon, such as this one shown here. And if we get unlucky, we might have an electrode that's right on top of a freeway of axons and makes a complete mess of the neural code when you activate it. In fact, we know very well from the second sight experiments and beautiful behavioral experiments that were done with them that part of a major problem in the visually evoked responses there was the activation of axons. So how can we deal with this? Well, we don't have a magic bullet for this, but we exploit exactly what I just showed you, which is that sometimes we do great and other times it's harder. So we just need to be able to figure out which electrodes work well and which ones don't, and I'll show you that now. The way we do this is to examine the electrical footprint of cells that are spiking and see how well that electrical footprint matches what the activity we evoke when we stimulate. So the electrical footprint of a cell firing is shown here. This is what we call the electrical image of the firing of a single ganglion cell that's obtained during visual stimulation. So it's with no electrical stimulation. If we average across many recorded spikes, this is what we see when this cell fires a spike. The spike initiates here at the soma of the cell, and there's the dendritic components surrounding that, and then the signal propagates up an axon down toward the optic disc. Okay, and if I showed you 500 cells from the same recording, they would all be sending their signals up this way, and in an image shown here, for example, 4 different cells of this same type. So this gives us a sense of the electrical footprint of the cell. What happens on the array when this particular cell fires a spike? Now we go into our electrical stimulation conditions, see, can we reproduce that? So here's an example of that, where we have the retina, the axon fibers running over it, the electrode array, and here's the electrical image of a particular ganglion cell that we recorded. Again, here's the spike waveform at this electrode. This is the somatic spike waveform, an axonal spike waveform propagating down the axon, everything exactly as we would expect biophysically. Now we go and we pass a small amount of current through a particular electrode, about a microamp of current through this particular stimulating electrode right here. And you see that the pattern of electrical activity that we evoke looks basically identical to this pattern of activity here. So we've created a spike in this one cell with this amount of current. Now, if we pass a little more current, unfortunately, we see a big mess. Not only is that cell firing a spike, but we see all this activity running up this way. And all the axons, of course, are going down in this piece of retina. So the only thing that this can be is the activity of axons that we have retrogradely activated by passing current at this location. So in this simple example, we see that if we pass 0.9 microamps of current, we're good. And if we pass 1.3 microamps of current, not so good. So that makes our choices clear here. Don't pass 1.3 microamps of current through this electrode, pass 0.9. Now, if we could do this all the time, that would be fantastic. We can. Sometimes the thresholds are favorable this way, and sometimes they're not, because we think the electrodes sometimes land closer to a cell and sometimes closer to an axon. And a summary of the electrical response properties like that is shown here with the electrical stimulation thresholds in 3 different retinas from somas, near the somas, and the axon bundle thresholds. The bottom line is, in about half of the cases, we activate axon bundles at lower current levels than the cells. That electrode is no good. In the other half of the cases, we activate cells at lower current levels than the axons. That electrode is good, and we can use it at a particular calibrated current level and know that we're evoking the correct amount of activity. So we don't have a magic bullet for getting rid of axon activation. Instead, what we do is we say, you know what, put our electrode array down, half the electrodes don't work, calibrate those, throw them out, don't use them, and then the other half, use them at the current levels that we know create electrical activity in a single cell without creating electrical activity in axon. So that's our approach to avoiding axon activation. Do you want me to take current questions as we go, or maybe
We can wait for the
we'll
end.
wait? Okay. If somebody has an urgent question, of course, please jump in if you need a clarification, and I'm happy to answer more questions. So this is how we detect and avoid unwanted axon activation. Last, but very much not least, I want to turn to approaching the central retina. As you know, the central retina is the locus of high resolution vision. That is the target for visual implants. And everything I've told you about in these slides so far has been in the peripheral retina, 6 millimeters or more from the fovea, which is to say 30 degrees or more from the foveal vision. Can we approach and target the central retina? So we want to be here near the fovea in the retina. This is an image of the blood vessels as well as the axons that run over the surface of the retina. There's a region that we've targeted here called the raphe. The fovea itself, as you know, is a very complicated structure with ganglion cells pushed off to the side right in the fovea, piled up nearby the fovea in a complex way, and so on. So we are not able to target the fovea now, and it will take time to get there. We can talk about that at the end of the talk. However, out in the raphe region, this is a particularly interesting region of the retina. And the reason it's interesting is not only is it close to the fovea, allowing us to march toward the fovea and see how well we can approach it, but also it turns out that the axons coming from the peripheral retina take a turn and avoid the fovea to go to the optic disc, leaving an area called the raphe or seam almost devoid of axons. That's super convenient for us because we don't like those axons. So it's a great target. And this is an image we took in my lab of these axons going around the raphe area here. It's a beautiful target for electrical stimulation. So we do target that area. And here's shown an electrode array that's at the same scale as this image shown here. It's not the same retina, but it's the same scale and targeting this region. And what you can see is that those electrical images of the recorded ganglion cells sometimes go up and sometimes go down. It's a very unique signature of this part of the retina because some of these axons go up and some of these go down. And there's no other part of the retina where that happens. So we know we've targeted the raphe to a resolution of about a 1/4 millimeter, something like that. So these are targeted recordings in the raphe area of the retina, approaching the central retina at about 2 to 3 millimeters eccentric, 10 to 15 degrees eccentric. Now, how well can we activate the cells there? Well, as a comparison, I'll just remind you, this is the same data I showed you before in a different format with a whole lot of red cells in the peripheral retina from 8 different retinas showing the degree of selectivity with which we can activate those cells. So the peripheral retina is pretty good for selectivity of activation. When we then perform these recordings in the raphe area, 2 to 3 millimeters, we get a picture that looks like this. Much less complete recording from the cells and a lot less red and a lot more blue. So what that's saying is that, yes, we can still sometimes selectively activate an individual cell, but we have more trouble doing so. Not surprising, there's a much higher density of ganglion cells in this area of the retina, so selective activation becomes more difficult. Still, we can do it a fair chunk of the time, and still, again, it's a completely different story from activating dozens or hundreds of cells of unknown types over an unknown region. This is a highly calibrated system where we know exactly what patterns of activity we're creating because we're able to record the activity of the cells in that region. So with an imperfect stimulation in the central retina and a better stimulation in the peripheral retina, what's the better target for the neural code and for recreating vision? Something more central or more peripheral? So to evaluate this, I'm now going to try to put together information. This is what you're looking at is a very reductionist view, if you will, of the degree to which we can activate individual cells. What does this mean for neural coding and restoring vision? So to get at that, I'm going to show you the analysis that we do of the capability to restore visual function in the raphe and compare it to the peripheral retina. And this is conceptually, oops, there we go. This is the most conceptually complex slide in my deck here, so I want to take you through this slowly to explain how we evaluate the potential for visual restoration, okay? So we've got our situation with our electrode array on the retina. We're stimulating, we're trying to create these different patterns of activity. And what we want to see is how well are we succeeding at creating the right pattern of activity. But in fact, we're less interested in the particular spikes than we are in what a person who has this device could see. So ideally, we would be fine-tuning our system to create the best possible visual image. How do we do that? Well, one way to do that might be if we knew what the brain was doing with the retinal image or had a nice model for how the brain works. Then we could come over here with our artificial retina and say, look, I have a model for what the brain is doing with this input. I'm going to turn my knobs on my device here that I've calibrated until I create a really good image. That's what we'd like to do. Of course, if we had a model for how the brain puts together visual signals from the retina, you would have heard about it by now, and somebody would have a Nobel Prize for that, right? But that doesn't exist yet. So what do we do in terms of thinking about how the retina is utilizing the incoming retinal signals to fine-tune the function of our device and then evaluate it? What we do is we create what I will call a surrogate model for the brain. It's not a serious model for what the brain is doing, but it's not a stupid model either. And we're going to assume that the brain performs an optimal computation on the incoming retinal signal, okay? I'm going to say, suppose the brain is taking those retinal signals and is doing the best it can to reproduce the individual image, okay? To reproduce the image that's incident on the surface of the retina. How do we do that computation? We can go into details, but we use linear decoders as well as CNNs to improve on the function of those. That's a little bit of a side topic. I won't go into it right now, but we build a surrogate model for the brain that, again, it's not a real serious model, but it's not ridiculous. That's an efficient brain. Then we say, great, now we have a model for what the brain is doing with this particular retina that we're recording from. We go and we calibrate this retina that we're recording from and stimulating. That is, we pass current through different electrodes, current levels, stuff I told you about before, figure out, avoid the axons, all that kind of good stuff. Do the best we can. Now we stimulate optimally, and that stimulating optimally, again, is a little bit of a technical side show that I don't want to go into in detail, but rather than stimulate simultaneously on many electrodes, we run around with a particular electrical stimulus on many electrodes, slightly out of sync with one another in order to avoid interactions. And we do this in the most efficient possible way using algorithms we've developed. And then finally, with all this business, our surrogate brain in place, our calibration of our retina, this is an experiment. So we're calibrating that retina. The surrogate brain we have is tuned up to listen to this particular retina. And then we stimulate optimally in this particular retina. We evaluate what's the quality of the decoded image that could be produced under this scenario. So I hope that makes sense, and happy to go through this again with you later. But let me show you what this looks like in terms of the actual data we record and how well we think we can reproduce the visual signal. So the data looks like this. Here's a collection of on and off paracel cells, two of the major ganglion cell types, our favorite two. Here's the visual stimulus. And what we know from this whole paradigm I just told you about, that if we were able to control the activity of every cell perfectly in that region of the retina, this image here is what the brain could reproduce. Okay, that's our goal if we had perfect control. We don't have perfect control. We have the control that we obtain from stimulating and recording and calibration and axon avoidance and all that business. And so how well can we do? Well, we calibrate, set it up, we optimally stimulate, and the optimal stimulation looks like what's shown on the lower left, and the image that's built up rapidly over 50 or 500 milliseconds is shown on the right. So what you can see is that as we run around and stimulate, the visual image that accumulates over an integration time of 50 or 100 milliseconds is shown on the right. It's not perfect, but it's awfully close to what we could achieve if we had perfect control. I'll play that for you once again. So we optimally stimulate based on all of our calibration, and we estimate what the brain could reproduce from that, and we get the image shown below. We can see that that's very close to what we could get if we had perfect control over the different cells in this region of the retina. Okay, so now how does the peripheral and central retina compare? So this is recordings from these are 8 different recordings from the peripheral retina. This is the image shown of ideal stimulation if we had perfect control of the collection of parasol cells in that retina. And here's what we get when we actually stimulate and see what we get from the cells. 8 different recordings shown here. The on and off parasol cells are used, and basically this is the degree to which we can reproduce this image. There's some structure that you can see there. It's not great. In the central retina, it's much better in the RAFE. So we have poorer selectivity, as I told you before, but we have access to more cells. And that balances out in our favor in the central retina. We get high quality reproduction of these images and close to what we expect from the ideal control of those different cells. So, and this is again, 8 different RAFE recordings, each one analyzed separately, each one stimulating as optimally as we can. And the quality of the image you can assess by looking at how well it matches the visual stimulus up here. And we can see a whole lot of the structure. It's significantly better than what we get in the peripheral retina. Naturalistic images can be done this way as well. We have a more fancy surrogate model of the brain to interpret these naturalistic images. We can talk about it separately, but the basic idea is still the same. In the peripheral retina, this is the natural image we're presenting. In the peripheral retina, we see that this is the best we can get from stimulating. If we had perfect control, but with the actual measured control, this is what we can get. It's pretty close to this, but it's all rather blurry because of the limited number of cells. In the central retina, we do better. So in these peripheral versus central recordings, moving to the RAFE, getting closer to the fovea, has improved our ability to reproduce the visual image. And we expect to continue moving toward the fovea in the future based on these results. So what I've told you about then is 4 major topics, and I did not discuss these 2 topics, which we could talk about in the Q&A, using the laboratory prototype to evaluate how effectively we can reproduce a neural code and deliver visual image to the brain. What I want to mention briefly as I wrap up is our efforts toward building an implantable device that can do all these things. Everything I told you about is isolated retina in a dish, in a laboratory, in a room full of equipment with 2 PhD students and a bunch of computers and all this. We want to boil this down to a tiny implanted device that can do all of these things. In particular, the things that our device needs to do, again, is to record the spontaneous activity, to figure out where are the different cells, what are the different cell types there, stimulate and record in order to see how well we can activate those cells, and exactly how much. And then finally, stimulate optimally to reproduce the neural code of the retina. Turning this room full of equipment into a tiny implanted device has a lot of requirements, all the way from electrical engineering of the chips, the material science of the interface, the surgical approaches, the packaging into a device that can be implanted and remain, and that's a large project. And the various different areas of this are shown schematically here for you to appreciate the different types of fields of science and engineering that need to come together to create a device that does all of these things. It's complicated, but we're working on it. And this is the Stanford Artificial Retina project that's doing these things. And I'm happy to answer more questions about that in the Q&A. What I want to finish with, however, is just to leave us with coming back to maybe the top level of the potential impact of studies, I hope, like this and studies of other studies of this kind. So first is obvious vision restoration. Our major goal was vision restoration, and that's simple enough. The device we're creating, we hope, will be a research instrument as well. In other words, a device that we can implant in experimental animals in order to test how the different cell types contribute to visual perception. And we have a project toward doing that. So it's really, I think, a spectacular research instrument for understanding the visual system function, because we get to dial in the control of the different cell types however we please and see how that produces vision. That in turn will help our clinical device, because we'll know better what the different cell types do and how important they are for vision. Once we're in that state where we're able to control the cell types and examine their visual function, then it opens up many questions about what I would call visual augmentation, which is the ability to deliver visual signals over and above the naturalistic visual signals that are present in our natural vision. The sky's the limit on this, and we really don't understand what that limit is at this time. What could we do if we could harness the bandwidth of the optic nerve and control it as we please in a much more complex and rich way than can be done in natural vision? We don't know, but we certainly intend to tackle those things. And then finally, I just want to mention that this is really a doorway into thinking about brain interface in general. The issues we face here in terms of taking an electronic device, implanting it in a circuit that's complicated, has many cell types. The cell types do different things. They send information to different places. They get inputs from different places. Every circuit in the brain is like this. I think it's a safe thing to say. Has all of these same problems. But we're able to tackle them now in the retina because we understand that much about how retina works, what the structure of the retina is, the different types of visual signals. And we're able to ideally use this to exploit the patterns of activity to do all the things that I told you about. I think in the future, these types of what we learn in the retina will translate in an interesting way into how we approach the interfacing to all sorts of brain circuits. So with that, I just want to end by thanking my collaborators, the Artificial Retina team, which includes people from my lab, PhD students and postdocs who work in a variety of realms from physiology to algorithms to development of devices and other aspects, a collection of crucial collaborators in all the different fields of study that I told you about, as well as others shown here. If you want to learn more about our project, this is our URL, and I'm happy to answer any further questions about it. Thank you for your attention today.
Chief.
Impressive, very, so a couple brief questions. What is so first I was confused. I thought you were gonna just focus on the peripheral retina, but the output that you're getting from the central retina, despite all because of the overlay of cells is much better. So because the peripheral retina by definition in the human means you're limited to 2200 vision or so, but from the central retina you have that's where 2020 comes from. What is the theoretical, in your mind, capability of such a device, and how many electrodes would it take to get to that upper limit?
Yeah, so great. So the far peripheral retina is very coarse vision, as you just said. Where we're recording now, we get closer. The thing that we're able to do in this part of the central retina, the raphe area, 2 millimeters or so, is we still have a monolayer of ganglion cells that we can access with relatively high efficiency using our flat electrode arrays. And although they're dense and we can't control them quite as well, as you just said, we can control them better enough that we can create a better visual signal. As we get toward the fovea itself, where we really would like to be, we have 5+ layers of ganglion cells at extremely high density. And then as you get into the really into the fovea, of course, there's no ganglion cells. So it's very messy, uneven terrain, not suitable for our flat devices. And how do we even access those 5 layers of cells? We don't have answers today, but I'll tell you what we're thinking. So first of all, we think we will need obviously curved devices, and that is produced by what I call an interposer, which is a non-flat surface that interfaces with the foveal area of the retina. That we think we can produce, and we have ways of doing that with new fabrication technologies that are sort of they're based on 2-photon imaging and basically fancy 3D printing, if you will, of these type of devices. In the multi-layer zone of the fovea, we're going to need to have very high densities, and probably we need to penetrate into the multi-layer zone in order to get through to the ganglion cells. To penetrate into the retina, first thing you got to do is you have to get rid of the ILM, the inner limiting membrane, which is a sheet that basically covers the retina. That can be done, as you know very well, by an ILM peeling procedure the retinal surgeons do all the time, and we've done it in the lab a bunch of times as well. So that's, I think, okay. But we need to penetrate the retina, and so we are developing penetrating electrodes using some unique technologies to do that with our material science collaborators. These penetrating electrodes are highly customizable, also with this fancy 3D printing type of technology. It's not ready to roll out yet, but we have nice prototypes in our first actual recordings and are submitting a paper on these electrodes now. So the way I'm thinking about it in terms of marching toward the fovea, if you will, and creating very high density electrode arrays and penetrating stuff like that, is that we start there in the raphe. We can get a pretty good visual image. It's at 10 or 15 degrees eccentric, which is still pretty eccentric. But we know that people can learn how to read and use that and move that around using the equivalent of a preferred retinal locus, right? And experiments have been done, and we are doing some in our lab as well to see how well we can perform in that area. That would be version 1.0 of an implanted device. Not what we're looking for in the end, 1.0. If we can really recreate the neural code effectively there and show that it works in humans, then we are very motivated to take the next steps of denser penetrating electrode arrays and a curved surface thing to move into the fovea. That's the plan. Did I answer it? Okay.
So the last bit of how big an array do you anticipate that being?
Right. So the other thing is the size. This is a tiny, the electrode array we have right now is small. It's 1 millimeter by 1 millimeter squared. Scaling up, and that's a very minimal thing. It's roughly the size, just a sense of it. If you hold your phone at arm's length, that's the kind of thing we can cover with a square millimeter. That's not a whole lot. But if we can make a nice image with your phone at arm's length, well, maybe you can see the image of your daughter's face on your phone. That'd be pretty good. So can we get there? We'll try. We think that an image that's maybe 10 times that in total electrode count, so 3 by 3 millimeters, that'll be a pretty good visual image. And then scaling more is, you can imagine all the issues. It has a range of issues: implanting the hardware, getting it into the retina in the first place, which I'm sure you can appreciate, holding it there successfully, heat dissipation, all the cables, and so you have to fit a lot more hardware into the retina. I think that will come. I think we start with our small device and see how well we do. Thank you. Thanks for the great question.
All right, let's move to Justin.
Hey, Jay, good to see you.
Justin, all right. Hey.
So I was struck, you spent some time talking about how you basically have to toss half the electrodes because they have an unfortunate location. You know, they're on an optic nerve or an axon bundle. I was curious, are there patterns of like electrical stimulation you can do that actually decrease the likelihood of an axon bundle or retinal ganglion cell firing? I'm thinking of like low current to desensitize or something like that. And would that be helpful?
That is a whole area of research in my lab that I didn't touch on at all. Everything I talked about today, thank you for that question. Everything I talked about today was stimulating through one electrode at a time and activating one cell or one axon at a time or whatever it is, and working with that. We in fact work on something many of you might be thinking of. What happens if you pass patterns of current? Can you steer the current in a way that is favorable and avoids this axon activation while stimulating cells? And we have a number of different studies on that. I'll mention briefly that a classic idea is if you have 2 electrodes on either side of an axon like this, and one is positive and one is negative, you create a situation where there's no second derivative this way, and therefore you don't activate the axon. In fact, that works to a degree. We can reduce axon activation with bi-electrode stimulation. We can also do tri-electrode stimulation to better steer the current onto various cells. We don't have a home run on this yet. It's not like we can go and radically change the dynamics with this, but we can fine tune our simulation to have small numbers of electrodes simultaneously and do better. A second approach is the penetrating electrodes themselves. These penetrating electrodes that are intended for use in the fovea, shown in this slide right here, if they're very short penetrating electrodes in the periphery, we can just go through the axon layer and get closer to the ganglion cell bodies. I think that's going to be probably the big home run, is getting past the axons in the first place so that we don't have to deal with them. And then you mentioned temporal stimulation properties. Are there fancy tricks we can play with temporal electrical stimulation in order to access cell bodies rather than axons? So far, we've found techniques that allow us to access axons better than cell bodies. And we have an abstract on that for NER this year. We haven't found temporal manipulations that allow us to do the reverse. Activating the axons instead of the cell bodies can be useful sometimes, actually. We can actually get a cell on its axon and not activate the soma that's nearby. That can help us in terms of selectivity, but again, it's not the home run. What we're really leaning into is the penetrating electrodes.
Cool, thank you.
And John.
This was fantastic, and I actually a little bit sci-fi for me. That's fantastic.
I like the sci-fi comment, and what I say to audiences sometimes is, yes, it seems very sci-fi, but it's actually just sci now.
It is, yeah, exactly. But then, so let me ask 2 questions. One probably more scientific, and the other even more sci-fi. So, first, if photoreceptors are present, a bit of them, how this would affect the whole system recording and basically the whole.
Fundamental question, particularly for AMD, when you're in the region where there's still some photoreceptor stuff. I don't know. So the beauty of the system, however, is that with a recording system there that can record the activity and figure out what's going on, we can understand what the natural visual signals are that are still flowing. And then we can work around that. And all of our calibration and algorithms, all the stuff that I told you about that stimulates optimally, well, we can re-optimize and say, look, here's what's flowing through the circuit. The cell over here is doing pretty well. The cell over here is getting nothing. The cell over here, we need to add a little bit of signal to it. So in principle, we can do quite well with that, but we haven't actually tried to do that yet.
And allow me this question. So have you thought about the system, having system a little bit more plastic so it can actually teach ganglion cells what they should see for example, through overexpressing, Yamanaka factors for a small pulse and can teach by showing many images or something like, I don't know,
Teach the ganglion cell using molecular manipulations.
for example, or also electrical.
I'm displaying my bias here in that I have thought about it in terms of electrically teaching the ganglion cells or their downstream targets. Because there is a question about what happens to the downstream targets. Have they been influenced through generations? You have to reteach the brain how to understand these signals that you're transmitting. And there's reasons to think that, certainly from literature on people who've had sight restored from cataracts and other things. So I think this is the cutting edge of the sci-fi. And if we can go in there and electrically tickle the cells in a certain way, can we do things? So, and here's a slightly more concrete example. What happens when we stimulate just the on and off parasol cells and none of the others? Can you create a visual image? We have no idea, because never in the history of humanity has anybody ever stimulated just the on parasol cells and off parasol cells and left all the other ones quiet. What kind of visual image will that produce? I don't know. Maybe the first time you do it, it's nonsense. Maybe over a little bit of time, you can start to understand that there's patterns of activity in these two cell types that are conveying a nice image and the others are quiet. That's a form of teaching, right? Can we separate out the different the recipient signals by changing the timing of different cell types in the retina, making them more distinct from one another, but in a controlled, systematic way, not just making a mess of all the activity, but gradually teasing things apart and separating out signals of different cell types? Don't know. What happens if you deliver two different visual images to two different cell types? Right? The example I like to give on this is it's tongue in cheek, so bear with me, but I think it's worth just conceiving of what the future could be. When you drive down the freeway and you talk on the phone, you can do this more or less safely because you're engaging two different parts of your brain. When you drive down the freeway and look at your text messages, you can't do this because you're creating conflicting information. What if we have cell types that convey the information about the moving cars on the freeway, probably the parasol cells, and we have a different cell type that conveys the fine-grained visual information about what you're reading, probably the midget cells. And we stimulate the parasol cells with the image of the incoming cars and the midget cells with the image of the text. Can you read and perceive movement and avoid collisions at the same time? Obviously, we would not want to do this experiment. It's not the goal of my research to do this, but conceptually, this is the kind of thing we could imagine doing. What would that even look like? We don't even know what that looked like. It's not going to look like the superposition of letters and moving cars, I don't think. But if you can process it separately, clearly it would look different from that. So I don't know, the questions to me are endless. And this is kind of why I described the research instrument as a really important part of this entire project, which is until we can go in and control the cells and do the right things with them and evaluate vision, we can't answer any of these questions. So that's the game. And the learning in the long run is a really big deal. If we look at the history of auditory implants, of course, cochlear implants, it started with people who are deaf in adulthood. And over time, we realized that implanting them in children was a huge game changer in order to develop. The diseases are different in the retina, of course, but basically tapping into our learning mechanisms with this device and figuring out how to use it. Love these questions. I'm sorry I have no answers, none, until we build this device.
Very well, we have many questions, and we have Daniel Palanker, who is going to make a final comments. So let's move to John.
Hello, great presentation. And my question, I feel like you've answered sort of here and there throughout the Q&A and talk. And my question is an extension of Dr. Palczewski's on the bipolar cells. Have you considered having the electrical stimulus be at a certain frequency or amplitude that mimics the firing pattern of a bipolar cell to really maybe even change the circuitry such that the ganglion prefer the connection to the electrical stimulus?
That is not something we seriously consider. There's a few reasons that so first of all, bipolar cells typically don't fire. Some of them may, but most of them don't. So they're graded potentials for the most part in the bipolar cells. The challenge there is that therefore recording their activity in any systematic way over any reasonable chunk of the retina is, current techniques, very difficult. So we don't even know what patterns of activity would be creating in the parasol cells. excuse me, in the bipolar cells. Not knowing what pattern you're creating is a real problem. And maybe you could patch onto one bipolar cell while recording for the activity of a bunch of ganglion cells. You could do that, and you could start seeing what happens if you stimulate that bipolar cell. But we won't, I don't know how to translate that into a device that we would ever implant in somebody, because we're not going to patch onto the cells in a significant number in such a device. It also involves technical challenges that are currently still in the future for us, penetrating into the retina, accessing a different layer of cells. That's what these kind of electrodes are about. So technically, it's way hard. I'm not opposed to the idea. So Daniel Blanker is here today, and they're doing beautiful experiments and have an actual implanted device through a company that they work with that stimulates the bipolar cells effectively with photovoltaic stimulation. And that stimulates bipolar cells in a way that we don't exactly know what it is, but they have some ideas. And that certainly creates visual perception. And so they're closer to doing that than we will be for quite some time. I think their approach is beautiful, and it's because it's much simpler. It exists in patients today, which is fantastic. Our device is kind of, futuristic, of course, and it will take longer than we're near human implants yet. We still are designing the device.
Good.
Fantastic talk. I really enjoyed it. My question is about the practical implementation of this. So and you have the calibration shown here. So in practice, you'll have to deal with some variation, possibly pressure in the eye, fluid status, maybe electrolytes. I was wondering how often you envision having to redo this calibration to adjust for those variations.
Right. So I think the biggest one that I worry about is movement. So if this device moves by 10 or 20 microns, we have to recalibrate the whole thing, because all of a sudden every electrode is seeing a different cell or cells, right? So holding it fast on the surface of the retina is crucial. We think that that's going to work by putting a certain amount of pressure on the surface of the retina and having the device, if you will, indent a little bit into the retina. We know from the second sight experiments that you can have a big indentation into the retina, and the thresholds for electrical stimulation remain stable for months or years. So it seems that the retina can accommodate a little bit of displacement distortion in order to sort of fix a device firmly against it. It's certainly critical in our experiments that the retina be right up against the electrode array, no space. The space constant for doubling of the threshold is approximately 10 micrometers. So if we're 10 micrometers away from the retina, the thresholds are all doubled and the current spreads everywhere, and it's a complete mess. So we have to be right up against the surface of the retina, and our device has to be able to accomplish that. Could there be additional challenges? I suppose there could. I think this is a place where the second sight stuff, although it didn't create a high quality vision restoration, really set forward a range of things that can work. You can have such a device, you can implant it, you can implant it reasonably safely for long periods of time. The thresholds remain stable, indent into the retina, all that. So I think the path is pretty good, but we have to develop good surgical implantation techniques for these questions. We'll work on that.
Okay, let's move to Zach and Sam.
Thank you so much for that amazing talk. So do you anticipate that some people have touched on kind of the basis of my questions already, but do you anticipate that there will be retinal ganglion cell plasticity or remodeling with the extended use of the electrode arrays? And would that enrich the quality of an image that you're actually projecting onto it?
Yeah, so I mean, certainly the degeneration will have occurred in the retina. A lot of degeneration remodeling will already occur. We know that from the work of Brian Jones, who's here today. And we can, the circuitry is different. The ganglion cells are there in large numbers, maybe 60% or something toward end stage RP or something in that order. That doesn't mean that the circuits in the brain are still all intact. We don't know that. So there's lots of remodeling that will have happened before we implant. And I think you're asking the question, well, what happens after we implant? Is there additional remodel?
And would you kind of need ongoing calibration to work for that?
It's a great question. Everything I've talked about assumes that the brain is in a static state. It's optimally reading out the signal that's present in the retina. And we tune itself and we deliver the signal that's best for the brain to receive. You're like, well, what if the brain is changing out from under you? Great. So yeah, that makes it more complicated, but also interesting.
yeah, even in degrees of like photoreceptor degeneration, if you have underlying disease,
Yep.
you know, that
So
might
I
actually
think
be.
this ties a little bit into your question here of can we somehow manipulate the signal in order to train the cells better? Could it be that providing a very regular signal to one cell type at a time helps to sort of clean up whatever the brain is interpreting? You know, maybe if the signals are all a bit messed up and confused in the brain, okay, let's just get the on-parasol cells, target them, hit them with some images for a while. Maybe the brain will plastic and wait to start to make sense of that signal. And then we target the off-parasol cells and stimulate them for a while. It can do that signal. I'm totally making this up. Why? Because we don't have a device that we can implant yet. So until we have that device, it's fun speculation like this, but unfortunately I have no answers.
Hi, thanks for your really great talk. I have two quick questions. One is, have you ever considered, like, when you're making this implant, also putting a chip into the brain to kind of see if your stimulus is getting to the brain and what kind of responses are being evoked there? And the second one is about the modularity and upgradability of this device. Say, if it's powered and processed by like an iPhone today, in 5 years that might be hopelessly underpowered and like outdated.
So first question, have we considered putting electrodes in the brain in order to see what's going on there? We certainly could, and we probably will do a degree of that. The reason it's not super exciting to me is that the ability to monitor complete populations of cells of various types in the brain is nowhere close to what it is in the retina. You get occasional cells here and there among the hundreds of millions of cells in the cortex. You might record 100 or a few hundred of those all those cells. You don't know what part of the field, you don't know what's being represented, what's missing in your recordings, because recording in the brain is just harder. So the problem is, I'm sure we could use that to evaluate at least, is a signal getting there? That would be fine. But I don't think we're going to get to the point where an electrical recording in the brain is going to tell us how good the visual signal is. It'll just tell us how good it is on 100 electrodes. So I think it's way more important to do behavioral experiments in macaque monkeys and in humans, both of which we're vectoring to our device, to see what's the overall visual sensation. And by the way, the brain is not just the visual cortex, right? There are a whole bunch of different targets in the brain that are used for different aspects of visual perception. We're not going to target them all at once to see. I would much rather do a behavioral experiment, is the short answer. And the second question was about the device and what happens as technology improves. This design of the device is such that the part that's in the eye itself is the bare minimum implanted. Why? Because you don't want to touch that once you implant it. What you want is that as much as possible, the hardware is outboard. So as we experience technological improvements, better processing, better wireless data, communication, all this external hardware can be updated at will. So that is absolutely the design of the device. But once we implant something and push on the retina, particularly if it has penetrating electrodes, we're not going to do it.
Adam.
So I have a naive question. So what would you need to acquire color vision in terms of electrical stimulation?
What would you need to acquire color vision? The short answer is we don't understand color vision very well, but I can tell you that I've studied it, and so I know how well we don't understand it. I did my PhD research on color vision. So the best we know right now is we think that the small bistratified ganglion cells, one of the 5 major types, is an important player in blue-yellow color vision. And the midget ganglion cells, the most numerous 2 ganglion cell types in the retina, because of their varying sampling of the L and M cones, are important for red-green color vision, if you will. So that aspect, the red-green color vision, we can tackle with the midget cells, except that we don't really know how they encode it, except through which cones they're connected to. And in a degenerate retina, we won't know which cones. So we would have to play and perform behavioral experiments to see what kind of visual sensations, what color visual sensations are created by stimulating different midget cells. That would have to be a behavioral experiment. The blue-yellow, I think we can do pretty well with the kinds of technologies I talked about today, because we know the target cell type, we have a pretty good idea of how they're encoding it, and we should be able to introduce color vision into that. I don't do it in the slides and talks and stuff like that, just for simplicity. So blue-yellow, I think we have. Red-green, the science isn't there, and to the degree that it is there, tackling it will require some more fooling around with behavioral feedback.
Tony, are you there? I think he disconnected. All right, and now highly anticipated, Daniel.
Okay, so I will make a brief kind of comment, not to keep everybody for long. First of all, we are with EJ in the same building across the corridor, so we discuss these issues very often, and we disagree on a lot of things, but we agree on one main issue, that all this technology should respect the retinal code. And we know this now from clinical results of many trials where retinal prosthesis or optogenetic stimulation was applied without respect to retinal code.
Left edge is not visible.
And patients.
Eric, please mute yourself.
Yep, sorry.
Okay. So patients saw light, but not form vision. And that's really, I think, the message that now we should really take seriously, that we should encode retinal information in a way that brain will be able to decode it and see as a form vision. As you know, we do it by going through bipolar cells and letting retina encode it, and our patients really see, have form vision, can read letters, and that's, I think, for the first time. And the effort that EJ described is exactly targeting the same kind of issue, how we properly encode information, but now skipping the bipolar cells and going after ganglion cells. So there are a lot of technical challenges that EJ described. Identification of retinal ganglion cell types in degenerate retina is a big one because their patterns of spontaneous firing is changing. Obviously, you don't have light-induced input, so you have to figure out from spontaneous firing despite the degeneration, but this might be possible. And selective delivery of retinal code to proper types of ganglion cells all the time, despite the movement of the eye, and you have to have very stable implant, as EJ explained, within microns of stability. The challenge of targeting peripheral retina or in RAFE is also a question of utility, basically practical utility of this, if the vision will be lower due to limitations in brain wiring to this peripheral retina. And it will not be used, let's say, for reading. What it will be the utility, maybe for ambulation, but if it is ambulation for RP patients, which is important, it requires probably large field stimulation to make it useful. But I think the key question, I would put this technical issues maybe aside. The key is really the assumption, the big unknown that we only will know when this goes into patients, is what will be the perception. Because as EJ explained, we have about 20 mosaics of different ganglion cells sampling the visual field in parallel and transmitting this information to the brain in parallel. And now you turn off all of these inputs and you just stimulate one type, let's say. What will brain do with it? All the correlations are wrong now. There is no input along other channels. There is only one, let's say, channel on ganglion cells, on parasol, for example, ganglion cells. Is this a percept that brain will be able to interpret as an image, or it will be some confusion and it will be just light perception? And then you can add, a few other channels, off channel, for example, or midgets. But at what point the correlations will become natural enough for brain to interpret as an image? And I think this is a very interesting question that we will know probably only during clinical trials. So I'm very much looking forward to see what will be the outcome and what is that minimal threshold of unnatural kind of code, how close it should be to natural for brain to be able to have a form vision. So I think with that I will stop and
yeah, thank you, Daniel. I just want to say that Daniel frequently disagrees with me, but I never disagree with him. And that's another way in which we disagree. So thank you very much.
Thank you very much.